Good? Okay. Hi, everybody. My name is Alaina Smith. I am a PhD student in Andrew Barton's lab at Scripps Institution of Oceanography. And I just want to say that I am very excited to be here and would like to thank you all for letting me talk about my research on how dispersal and temperature variability influence phytoplankton realized temperature niches. So I'll first start out by saying that the ocean is a highly dynamic environment. Variability across regions and through time influence patterns in phytoplankton biogeography. We can see here in the Darwin model that as the model moves forward through time and environmental conditions fluctuate, the community structure and dominant phytoplankton types shift. Phytoplankton play important roles in biogeochemical cycles and food webs, which are heavily influenced by community dynamics. Different phytoplankton species and functional groups utilize different traits to influence their fitness and their ability to compete with species across environmental gradients. One notable environmental gradient that is highly dynamic in the environment influences key phytoplankton traits and is expected to see large shifts in gradients with impending climate change is temperature. So across all phytoplankton species, as temperature increases, maximum growth rate, a key trait for phytoplankton competition, increases exponentially. However, individual phytoplankton have unique temperature niches where so within these unique temperature niches that phytoplankton have, each species has a unique temperature where their growth is maximized, called the optimal temperature, and a unique range of temperatures where they have positive growth rate, called the niche width. Now, these curves are called the fundamental temperature niche, which are derived from laboratory growth rate experiments. So on the right, I have an example of the fundamental temperature niche for two ecotypes of Prochlorococcus using real laboratory experiments of temperature versus growth rate. And we can see here that for the ecotype MIT 9312, this one has a warmer optimal temperature. Now, fundamental temperature niches drive observed patterns in phytoplankton distributions in the ocean. So on the right, I have abundance cross-section across the latitudinal gradient in the North Atlantic or across the Atlantic for the same two ecotypes of Prochlorococcus. And we can see that the ecotype MIT 9312 that had the warmer optimal temperature is the one that is found in warmer low latitude surface waters. Now, we would expect that if the fundamental temperature niche is the full range of temperatures where an organism can grow, then the realized temperature niche would be constrained by these physiological parameters. However, we find evidence that the realized and fundamental temperature niches are different. So in this figure on the right, I am showing you a figure from a previous study of mine where I was looking at fundamental versus realized temperature niches for 4 ecotypes of Prochlorococcus. So on the x-axis, I'm showing niche width, and on the y-axis, I'm showing optimal temperature. The black dots are the parameters from the fundamental temperature niches derived from laboratory experiments, and the colored triangles are the parameters from the realized niches from observations split into different ocean basins. And so what we found was that on average, the realized temperature niches were colder and wider than the fundamental temperature niches. Now, why might that be? There could be a variety of reasons which could explain why we're seeing these patterns, but I am going to be talking today about the role that mass effects, spatial and temporal mass effects, play in influencing phytoplankton distributions and affecting realized temperature niches. So mass effects are a paradigm explored in meta-community ecology, aiming at explaining why we find phytoplankton in high abundance in regions outside of their preferred environmental regimes. And the mass effects are split into two components. The first is the spatial mass effects, which is the role that physical dispersal plays in pushing organisms outside of their preferred environmental regions. And the second is the temporal mass effects, which is the phenomena where we find organisms in high abundances in a location, even though they might have net negative growth rate. So an example of this could be bloom dynamics, where we can find species in high abundance, even though the co-located environmental conditions are not the conditions that initiated the bloom in the first place. So that brings me to my driving question, which is how does dispersal, which is modeling the spatial mass effects, and temperature variability, trying to model the temporal mass effects, influence phytoplankton realize temperature niches? So to answer my question, I created a simple meta-community model where 45 phytoplankton species, each with their own unique temperature niches, compete for the same limiting resource. Here, each species has their own unique optimal temperature niche, optimal temperature, which is each one of them is separated by 1 degree Celsius along a temperature gradient. They each have the same niche width, and the entire phytoplankton population dynamics are influenced by temperature-dependent growth rate, which is their fundamental temperature niche, Michaelis-Menten nutrient uptake kinetics, and then is balanced by temperature-dependent mortality and dispersal of population in and out of the community. So temperature, I'm using temperature-dependent mortality here to model how temperature affects the metabolic rates of grazers as well as phytoplankton. So temperature-dependent mortality has been utilized in models before, but the exact scaling factor of that is still sort of unknown. So in my experiments, I'm also going to be testing how different scaling factors of temperature-dependent mortality and therefore how increased grazing pressure influences these dynamics. And then finally, the population is influenced by dispersal in and out of the community. So some fraction of the population immigrates to adjacent communities, and some fraction of the population from those communities are immigrate into the community in question. So I have a latitudinal transect north to south, divided into 1 degree latitude wide boxes, each with their own unique seasonal temperature variability influenced from NOAA climatological data. And dispersal between boxes is dependent on average horizontal diffusivity and the distance between any two boxes. So this means that boxes closer together have a higher rate of dispersal versus boxes further apart. It's been estimated in the ocean that horizontal diffusivity can be as high as 10 to the 4 meters squared per second. And so in order to test how increasing dispersal influences my community, I'm going to test 10 to the 0, 10 to the 1, 10 to the 2, 10 to the 3 meters squared per second as my horizontal diffusivity constant. So all 45 species are seated into each box and allowed to compete for 50 years. After 50 years are up, I determine which species converged in the model if the net population growth over the last 5 years is equal to 0. And I also calculate if the species are in the model are sort of present in that community by saying if the abundance in the last year is greater than 10 to the negative 4 times that of the most abundant species in that box. So I'm going to sort of walk through my the conceptualization of my experiments, and which is going to be on the left. And then on the right, I'm going to have an example of the model output. So on the right, I have the first column, which is just seasonal temperature for 4 different boxes. And then in the second column, I have the biomass for all of the surviving species in those boxes over the same year. So for the first experiment, I have constant temperature and no dispersal between boxes. So each species has the same niche width in their fundamental niche, only differ by their optimal temperatures. And in this experiment, I'm sort of testing competitive exclusion. So if there's no temperature variability and no dispersal, then I'm expecting only one species in each box, which means that there won't be a really calculatable realized niche because temperature is constant. And so from this experiment, in the example model output, that's what we can see, only one species present in each box. And the species is the one whose optimal temperature, that's what they're colored by, is closest to the mean temperature of the environment. My second experiment is now keeping temperature constant and allowing for dispersal between boxes. And this is me testing the effects of the spatial mass effects. So the fundamental niches are the same, but now that species can disperse between boxes, I'm expecting for the realized temperature niches to widen as they move along the gradients. And we can see in the example model output for the same boxes from the competitive exclusion example that we now have more species present in each box. So the dispersal is physically pushing these species around along this gradient and resulting in visible increased diversity. The third experiment is now allowing temperature to vary naturally in each box, but for no dispersal between boxes. So if the fundamental niches are still the same, I'm expecting for wider realized temperature niches, but I'm actually not expecting for temperature niches to be as wide as the dispersal case, just because there's no dispersal between boxes, so it's gonna be just dependent on the variability in that box. So the example model output, we can now see that each box has a distinct seasonal temperature cycle, and that output of the community, we can see that the species are cycling in the same dynamic, like seasonally, as the temperature optimums shift over the season. So different species take advantage of the temperature regimes at different times of the year, which increases diversity in some boxes with really high temperature variability. Now, my final experiment is to combine these two and now have natural temperature variability in each box and allow for dispersal between boxes. So now we can see in the example model output that diversity increases greatly because now not only are the boxes being influenced by the seasonal variability locally, but also the seasonal variability in adjacent boxes increase diversity in those boxes, and dispersal pushes all of these organisms to interact with each other. So now I'm going to walk through specific results from each of these different experiments. On the top right, I put up the little schematic of my model hypotheses, just as a reminder of what experiment I'm talking about. So the first is I'm talking about the spatial mass effects, which was when temperature was constant, but dispersal was allowed between boxes. So in this, I'm looking at, on the x-axis, I have increasing dispersal magnitude, and the y-axis is just all of the different boxes, and they're colored by the average diversity in the last year of the model. And so this is an experiment where mortality is lowest. And we can see that as dispersal increases, diversity increases at all latitudes. And when when dispersal magnitude is really high, we actually get this sort of everything is everywhere. And now immigration from other boxes is greater than the loss from mortality. However, when I increase mortality, I still get increasing diversity with increasing dispersal magnitude, but at the extreme dispersal magnitudes, at the highest dispersal magnitudes, I'm no longer getting this everything is everywhere effect. And in extreme environments, the temperature-dependent mortality is now balancing and outcompeting the force of immigration from other boxes. And so now I'm going to talk about the influence on realized temperature niches specifically. So in panel A, I have dispersal magnitude on the x-axis, and on the y-axis, I have the ratio between the realized niche and the fundamental niche width. And so values greater than 0 mean the realized niche width is greater than the fundamental niche width. So we can see here that as dispersal magnitude increases, realized niche width increases as well. In panel B, I'm looking at dispersal magnitude still on the x-axis, but instead of niche width, now I'm looking at optimal temperatures. So values greater than 0 mean that the realized optimal temperature is greater than the fundamental optimal temperature. So what we can see in general is that as dispersal magnitude increases, realized niche width increases, but there's no significant effect on optimal temperatures. And we can also see that as mortality increases, which are the different colors here, the orange at the top is the lowest mortality, and then as mortality increases, this sort of effect is dampened, where now things are dying before they're able to reach that full extent and allow their realized niche to get as wide as under low mortality conditions. So now I'm talking about the results from the experiments where there's no dispersal between boxes, but natural temperature variability is allowed. And this is looking at the temporal mass effects specifically. So in panel A, on the x-axis, I have temperature amplitude. And on the y-axis, I have average species diversity in the last year of the model. And in panel B, on the y-axis, I have total species, so the total number of species present at any given time in the model. And we can see that as temperature amplitude increases, average and total diversity increases. Now the colors again are looking at the different mortality treatments. And in orange, the highest, the line that's at the top there is the lowest mortality. And so as mortality increases, again, we get a dampening of this temporal mass effects as species are dying before the seasonal temperature variability can return to an optimal window. So now we're getting less species diversity in boxes. And so again, I'm looking at the influence of the temporal mass effects on realized temperature niches. So in the left panel, panel A, I have temperature amplitude on the x-axis and the ratio again between realized niche width and fundamental niche width on the y-axis. So again, values greater than 0 mean the realized niche width was wider than the fundamental niche width. And then on the panel B on the right, the y-axis is looking at optimal temperatures. So overall, we can see that as temperature amplitude increases, realized niche width increases, but there was no significant effect on optimal temperatures. And we also have the influence of mortality. So as mortality increases, again, the temporal mass effect slightly dampens this pattern, and we're still getting an increase in realized niche width with temperature amplitude, but not as wide as when mortality is lower. So now I'm going to talk about my final experiment, which was looking at the combined spatial and temporal mass effects. So now temperature is allowed to vary naturally and dispersal is allowed between boxes. So I'm looking at diversity in this section. So the top row is average species diversity throughout the last year in the model, and the bottom row is total species diversity. So the total number of species that were present at any point in the last year of the model. Each column is the different mortality treatments. So as we move from left to right, mortality increases. And we can see that no matter what mortality we're looking at, as dispersal magnitude increases, the number of species increases, but we're also getting the effect from the increasing temperature amplitude. So in the northern hemisphere, the model actually has a higher average temperature amplitude compared to the same latitude in the southern hemisphere. So we can actually see that pattern start to emerge, where in the northern hemisphere, for the same latitudes, we're getting slightly higher diversity compared to the southern latitudes. And so my sort of pièce de résistance here, I'm looking at how the combined spatial and temporal mass effects influence phytoplankton realized temperature niches. So on the right, I'm showing dispersal magnitude on the x-axis. And again, that ratio between the realized and fundamental niche on the y-axis. But now the points, which are individual species, are colored by the total range of temperatures that they experience throughout as they're getting pushed into different adjacent communities. And so looking at the total influence of temperature amplitude. So we can see that, the total across all experiments, we see that as dispersal magnitude increases, realized temperature niche widths increase. But we can see even when dispersal magnitude is low, if you have a higher temperature amplitude, so looking at the furthest left cluster of points here, this is the lowest dispersal magnitude, but we're still able to get realized temperature niches that are wider than the fundamental temperature niches, so in this case, greater than 0, when temperature amplitudes are high. And so these combined effects are creating really wide realized temperature niches compared to fundamental temperature niches. And like previous experiments, we did not see a significant effect on the optimal temperatures with dispersal magnitude or temperature variability. So this brings me to some summary points that I sort of want to point out. First, independently, spatial and temporal variability increase diversity and widen realized temperature niches with no significant effect on the optimal temperature. And the effect of dispersal and temperature variability is dependent on the strength of mortality. So here, the temperature-dependent mortality I was using as a proxy for understanding how increased grazer strength affects the system. So stronger top-down control, where higher rates of mortality sort of diminish these mass effects. So in the case of the spatial mass effects, the rate of mortality is greater than the rate of dispersal, resulting in reduced connectivity. And the temporal mass effect, increased mortality, reduces the time that species have to survive between windows of favorable temperature. And then finally, the combined influence of dispersal and temporal variability results in realized niches that are consistently wider than fundamental temperature niches. And sort of to round it out with some broader implications, I would say that the temporal mass effects, from what I found, would be really important in highly variable seasonal regions, especially during times of the year when predators are least abundant. The spatial mass effects, on the other hand, would be most important in regions of strong advection and mixing, like the Gulf Stream, for example. And the mass effects combined would influence the entire meta community by benefiting regions downstream of those with high temporal variability. So in highly seasonal environments, diversity would be high, and dispersal would be pushing that diversity downstream towards regions where temporal variability might be lower, and therefore diversity might be lower. So just would like to acknowledge my funding agencies, the Barton lab members. Most of all of us are here. Check us out later, posters and talks. And thank you to the organizers for allowing me to speak, and you all for listening. And I will just say thank you and leave you with a little take-home message.
Thank you for that very exciting project. Thank you very much for that. Do we have questions? I guess we'll wait until, can I see who's online? Anyone has. I had, actually there was two. One was a clarifying question. Right at the beginning, you had the different sample boxes that you were showing, and you had different colors for different species. There were some that were dashed and some that weren't, and I wasn't sure what.
So the ones that were dashed were the species that were the ones that survived under the competitive exclusion example. So those were still the species that in subsequent experiments end up, they're still the ones that the optimal temperature is closest to that of the mean environment. So they typically have the highest abundance. So I was just showing that they still stick there. It's just other species coming in that increase the diversity.
Okay. And the other, another question, so the mortality you had as a constant, and it's sort of representing all sorts of other things. If it was grazers, there might be a temperature dependence of the mortality. And I just wondered if you'd thought about that, and if it would, would there be substantial changes, you think, to the results?
I think there would be some slight changes if I allowed for, if I had a better constraint on the actual grazing pressures, because I'm using temperature dependent mortality, which could be sort of as temperature increases, metabolic rates increase, and there's sort of like a natural death within that. But I was just trying to extrapolate towards how that influences grazers as well. But I think there would be, I think things probably would be slightly different if I actually included some constraints from
some actual grazers.
Yeah.
Anyone? Okay.
Thank you for the talk. I was just curious, you were for the species that you were just using richness, right? And I was just wondering if you had used something like a Shannon index or something that also weights it by abundance, if that would, I'm just curious how that would change the results. And I was just curious if you had looked at that.
So I hadn't looked at specifically calculating Shannon diversity index, but I kind of tried to get at that by creating that the constraint in the beginning by choosing which species were actually sort of present in the community as a result of their abundance. So because this model ends up as a steady state, all species are in each box and therefore have some abundance value associated with them. So I think if I utilize the Shannon diversity index, I don't think I would see much of a difference compared to just using the just total number of species that I was using here, richness. But I would, depending on how I would take into account which species are actually sort of like present in the community. But that might be a better metric to use. It's a good point. Thank you.
I was going to switch to an online question. Jonas, you had your hand up. You don't anymore?
Yeah, no. I think I worked it out in my head by myself. Thank you.
Anyone else here with a question?
That was great. I have a sort of a clarifying question, and then just want to get your thoughts. You show how the temperature variability, so the temperature amplitude, didn't really have an effect on the thermal optimum. And I'm wondering if how you were like assessing the thermal optimum. Is it at a specific point in the year? Is it an average for across the whole year? Is it an average of the species? And whether you looked at like the total thermal optimums across a year and whether that shifted at all?
Yeah, great question. I didn't look at, I was taking into account the entire last year of the model across the whole domain. So I was calculating the optimal temperature as the sort of central tendency of all of those points. So sort of the mean of this, of the curve, if I fit, sort of a non-parametric curve, found the mean of that, and then the niche width was just the difference between the first and the 99th percentile. So it was just the mean of the whole across the whole domain.
Any more questions?
I enjoyed your talk. I noticed that your model seems symmetric about the equator. Have you looked at possibly not seeding the southern or northern half of the model and seen how things invaded or dispersed into that southern half with only seeding on the north?
I hadn't looked at I hadn't looked at only seeding on the north and seeing how that translated towards the south, but I had run some other experiments where I sort of like flipped the temperature gradients of the north and south, and I was just kind of making sure that it wasn't just a geometry of the model, and I just essentially found that it the dynamics flipped. But it's also a good point to think about.
Well, thank you very much. So let's give another round of applause.